Hello and welcome to some biology class. Innate immunity is the defense mechanism that an individual is born with. It provides the first lines of defense against pathogens. Most of its components are present before the onset of infections. As a result, the response is immediate. It is less specific. Different factors that are associated with innate immunity are divided into 4 different groups. They are physical and mechanical factors, biochemical factors, cellular and genetic factors. First, we shall discuss about physical and mechanical factors that include skin, mucous membrane, cilia, coughing and sneezing, peristalsis, saliva, tear and urine. First one is skin. Skin is the largest organ in the body. It covers body's entire external surface. Skin serves as a barrier to invasions by microorganisms. Its uppermost horny layer, called as stratum corneum, is impermeable to foreign substances, as a result of which skin effectively prevents the entry of microorganisms into the body. But whenever the skin is lost due to burn or any other injury, pathogens easily enter the skin. As a result, infection occurs. Second one is mucous membrane. Mucous membrane lining various openings and passages in our body secrete mucus. Mucus, because of its sticky nature, trap pathogens. As a result, they fail to move. Mucus also prevents the adherence of microorganisms to the underlying epidermal cells. Third one is cilia. The epidermal cells of the respiratory passage are lined with cilia. The microorganisms that are trapped in the mucus of the respiratory passage are swept away by the constant movement of cilia. Mechanical actions such as coughing and sneezing are also part of the innate immune system. These actions drive out microorganisms that enter digestive and the respiratory tract. Next factor is peristalsis. Sometimes by escaping various barriers, microorganisms may enter the intestine. These organisms are pushed away by the peristaltic movement of the intestine. Next factors are saliva, tear and urine. Mouth is constantly bathed in saliva, as a result of which foreign substances that enter mouth are swallowed and they are digested in the gut. Conjunctiva of the eye is freed of foreign particles by flushing of the lacrimal secretion called as tear. In addition, the washing action of urine removes microorganisms from the urethra. Now we shall discuss about the biochemical factors that are associated with the innate immunity. First one is secretions of the skin. Sweat is a clear, salty liquid produced by the glands in the skin. High concentration of salts in the sweat has microbicidal activity. In addition, sweat is also slightly acidic, and this acidity also has microbicidal activity. The secretion of the sebaceous and sweat glands serve as antiseptics, as they contain lactic acids and fatty acids, which has bactericidal and fungicidal activity. But certain areas of our body, such as soles of the feet, are deficient of sebaceous glands. Because of this, these areas are easily attacked by fungal growth. Hence, these areas are known as alkaline gaps. Second factor is secretions of the digestive tract. The high acidity of stomach also has microbicidal activity, and this is due to the secretion of hydrochloric acid by the oxygenatic cells that line the stomach. Third factor is human milk, which is rich in antimicrobial substances such as lactoferritin and neuraminic acid, which fight against E. coli and Staphylococcus. And next one is nasal secretion and saliva, which contain mucopolysaccharides that inactivate certain viruses. Next biochemical factor is lysozyme, which is also known as N-acetylmuramidase. Lysozyme is present in tear, nasal secretion, saliva, polymorphonuclear leukocytes, human milk and other tissue fluids except cerebrospinal fluid and urine. Lysozyme break down peptidoglycan. Peptidoglycan, also known as murine, is a major component of gram-positive bacterial cell wall. By breaking down peptidoglycan, lysozyme compromise the integrity of bacterial cell wall. As a result, bacteria are lysed. In the presence of additional serum complement factors, lysozyme also inactivates certain gram-negative bacteria. Next component is interferons. Interferons are signaling proteins produced by cells in response to the presence of viruses. They are antiviral agents which inhibit intracellular viral replication inside the cells that are infected with viruses. These proteins inhibit viral mRNA transcription, thereby disrupting viral life cycle. Interferons also increase the activity of natural killer cells, thereby providing immunity to cancer. Because of anti-cancer and antiviral properties, interferons are used in the treatment of viral infection and also in cancer chemotherapy. Large amount of interferons are produced by recombinant DNA technology, in which the gene encoding interferon is introduced into E. coli where interferons are produced. Commercial production of interferons by recombinant DNA technology is a major breakthrough in science. Complement system is another biochemical component of innate immunity. Complement is a complex system of enzymatic proteins found in serum. They are activated by antigen-antibody complexes. Activated complement components increase phagocytosis of pathogens and thereby their destruction. Complement system operate by three pathways. They are classical pathway, alternative pathway and the lectin pathway. The main function of the complement system is the opsonization of microorganisms and formation of membrane attack complex which cause lysis of the pathogens. Next factor is properdin. It is a plasma glycoprotein encoded by CFP gene. Properdin along with complement and magnesium ion cause the lysis of gram-negative bacteria. It also inactivates certain viruses. Next factor is secretions of bacterial flora. Bacterial flora of the skin produce antimicrobial substances such as bacteriocins and acids. In addition, bacteria live in the human gut as commensals. These microorganisms also produce antibacterial substances such as colicins and acids, which prevent the entry of other microorganisms into the body. When these microorganisms are removed by antibiotics, pathogens get an easy access. In addition, lactobacilli of the vagina also produce antimicrobial acids, and finally, the mucous membrane also secretes antimicrobial substance which inhibit the proliferation of pathogens. Next factor is semen, which also contain various components such as spermine and zinc, and finally acute phase proteins, which are a class of plasma proteins whose concentration either increase or decrease in response to inflammation. One example is C-reactive protein, which binds the C protein of pneumococci. The concentration of CRP increase in response to inflammation. It acts as opsonins. Bacteria-bound CRP activate the complement system, thereby facilitating their uptake by phagocytosis. The cellular factors that are associated with innate immunity are phagocytes and natural killer cells. First, we shall discuss about phagocytes. Phagocytes are the specialized white blood cells which are capable of engulfing and digesting foreign substances, including pathogens. The process by which these blood cells engulf and digest foreign substance is called as phagocytosis. Phagocytosis was discovered by Russian zoologist L. Metchnikoff in 1838. He is known as the father of innate immunity. L. Metchnikoff along with Paul Ehrlich were jointly awarded Nobel Prize for Medicine in 1908. Two types of phagocytic cells are associated with innate immunity. They are macrophages and macrophages. Macrophages are polymorphonuclear leukocytes. It includes eosinophils, neutrophils, and basophils. These cells are short-lived and they do not divide. These white blood cells have distinct morphological characteristics. For example, in neutrophil, the nucleus is multilobed, whereas in eosinophil and basophil, the nuclei are bilobed. Out of these white blood cells, neutrophils are the most abundant white blood cells in the circulation, accounting for 40 to 70% of total leukocytes. Second type of phagocytic cell is macrophages. Macrophages can be free or remain fixed in a particular type of tissue. They are cells of the mononuclear phagocytic system, which was originally termed as reticuloendothelial system. Macrophages differentiate from bone marrow promonocytes. Promonocytes in course of time undergo differentiation and gives rise to monocytes in the blood, and monocytes after further differentiation gives rise to macrophages. After differentiation, macrophages can remain free or migrate to different tissues where they get fixed. Examples of free macrophages are alveolar and peritoneal macrophages. In addition, free macrophages are also present in lymph and other tissues. Examples of fixed macrophages are Kupffer cells in liver, osteoblast in bone, and fixed macrophages of lymphoid tissue and other tissues. So both macrophages and macrophages are professional phagocytes. They engulf and digest foreign substances such as pathogens and also cellular debris. Macrophages are long-lived. They are found engulfing bacteria, viruses, and protozoans. Another cellular component of innate immunity is natural killer cells or NK cells, which are characterized by the presence of surface marker such as CD56 and by the absence of CD3. They account for 5 to 20% of the circulating lymphocytes. They contain large granules, hence NK cells are also called as large granular lymphocytes. They exhibit natural cytotoxicity. Natural killer cells kill a wide range of tumor cells and virus-infected cells. Their response to the presence of virus is rapid. Natural killer cells differentiate from CD127 plus common innate lymphoid progenitor, which is downstream of the common lymphoid progenitor from which B lymphocytes and T lymphocytes are also differentiated. NK cells differentiate and mature in bone marrow, lymph nodes, spleen, thymus and tonsils. Natural killer cells are part of both the adaptive and the innate immune system. Natural killer cells have several functions such as lysis of virus-infected cells and tumor cells. In addition, virus-infected cells secrete cytokines which activate natural killer cells. Activated natural killer cells produce gamma interferons which activate macrophages. Activated macrophages then cause the phagocytosis and lysis of virus-infected cells. So friends, this is all about the cellular components of innate immunity. For more updates, please subscribe my YouTube channel, Sam Biology Class.